So that's a long time. And so I'm going to talk about plant phenomics data management challenges and some of the solutions that exist, especially around MIAPPE, the minimum information about plant phenotyping experiments. Those are especially the solution used at INRAe as well as ELIXIR and EMPHASIS. But those are not the only solutions. And keep in mind that although MIAPPE or breeding API compliant, exist currently in the research communities. So very quickly, because you don't necessarily know what ELIXIR and EMPHASIS are. So ELIXIR is a European research infrastructure for life science data from human to biodiversity and plant research. And what we want to do in the plant community is to build a sustainable tool federation. So far, we rather succeeded. Mainly aiming at fair data management through the adoption of standards in plant sciences to develop also community recommendations, as well as data portal. We want, therefore, to facilitate data integration and analysis, provide a lot of training, and help the building of joint projects. So I'm leading this with my two colleagues, Sebastian Bayer and Christina Gruden, from Germany and Slovenia, respectively, and with the help of Katrina Hai. And we have also a long history of collaboration with EMPHASIS, the infrastructure for plant phenotyping at the European level. It is also a network, and it has a French node, which is Phenome, where I am involved. And among the many activities of the EMPHASIS plant phenotyping infrastructure, there is one around data computational services, which has 10 years of history of collaboration with ELIXIR, mainly, but not only, around MIAPPE. So the plant phenotype management and publication challenges, it originated from the analysis that genetics, genomics, and omics data are mostly centralized, mostly homogeneous, even though the metadata, the description of the data, can be heterogeneous. But on the other hand, phenomics data are dispersed, will remain dispersed. There are many data repositories and databases hosting plant phenomics data, and this is going to last. Furthermore, the data, phenomics data, are heterogeneous, and they are more than getting standardized. They are mostly standardized now. And the environment data are very linked to phenomics data. The objective with all of our activities is to enable the integration of those different fields of research using FAIR principle to be able to do plant breeding, so genetic variations by trait studies, climate change studies, through genotype by environment or agronomic modeling, and so on. So what we have seen along those past 10 years is that when we talk about phenotype, we speak, in fact, of many, many different things. This is linked to a lot of things, and I cannot explain our position in only 20 minutes, but one of the key aspects is the phenotype data life cycle, which is quite complex. With a first step of data acquisition of raw data with phenotype and environment measurement with variables, where we see here this symbolizes a field with microplot where certain varieties, Soissons, Chargeur, are studied with different cultural practice, high or low nitrogen input, for instance. And what is commonly done is that those plot level data are derived, reduced, simplified through data computation to produce indicators, simpler data ready for integration. This also includes the transformation, the extraction of traits from sensors or from images, as seen here. This allows from those derived data to extract knowledge, which then get published. Those knowledge is, for instance, that variety shower resistance to fusarios and their intensive cultural practices. And what we can see is that the last one, the knowledge part, is the one that get mostly published. But if we cannot get back to the derived data, and more important, to the data acquisition level, to the raw data, we cannot reproduce all of this. And furthermore, we see that the raw data can be derived into many derived data sets. And therefore, it is very important to keep on the long term the raw data available in data repositories. So for data acquisition, it's variable, raw measures, phenotype, and environment. As I said it includes data cleaning. We must set up traceability, reproducibility, and provenance. And by provenance, we mean not only stating what methodology of measure, but also the methodology of transformation and the sensor identification and the devices identification. As I said, for the indicators, for the derived data, new computation for each scientific question. And therefore, one experimental data set can lead to many derived data sets. So there are several tools and solutions that are available alongside this phenotype data cycle. We need first planification tools that include genetic resources management systems that allows to release the collection, to get access to the collection, and so on. And platform information systems that allows to set up the experimental design and to prepare the actual experimentation either in greenhouse or in the field. We need then the actual data collection through portable devices, sensors, and this can be done by dedicated tools such as Adonis in France, but there is also a system within the breeding API community. Scripts and workflows, which are very community dependent. And finally, the very important part of preservation sharing, which goes through both data repositories, such as Dataverses, through the Recherche DataGov instance, on data portals, such as Feeder and Florilege. I'll get in more details on those. Those are just a little bit of examples of potential solution. The important thing here is that to get a link, a glue between all the steps of this life cycle, we need compatible data standards. And if we need compatible data standards, I even put, again, a famous cartoon on how we can go from 14 standards to 15, but we need to have registries showing what are the standards that should be used. And one I like personally is fair sharing that provides, among other things, collections, such as here the plant research data resource for European infrastructures that lists the potential standards, databases, policies could be of interest for plant researchers. This is also backed by a community driver recommendation and registries. What we have seen in the WheatIS community, so the global wheat initiative community, is that it has been very useful to set up pages listing the data standards, the data management practices. And this went one step beyond 4 years ago, or 3 years ago, through the RDM kit, Research Data Management Kit, available at this URL, that provides different entry points linking to data management practices. And this is really, and I'll get back to it later, a tool I really advocate to use because this will list you all the details, all the trainings that you might be using in your work. So the RDM Kit is a Wikipedia-like knowledge-based website. It's free, it's open. It's described how to manage research outputs according to FAIR principles. And it's a portal to other online resources. The idea, once again, it's not to centralize everything, but to give a link to the right resources used by RDM professionals and researchers. And once again, you've got the RDM kit URL here. One other important thing is that it's one of the resources recommended by the Horizon Europe program guide. What it's not a peer-reviewed journal. It's not a repository to store file. It's not a data repository. It's not a simple registry of resources and tools. It's not a manual or user guide for tools. It's not a website for projects to share deliverables. RDM Kit works because there are many people behind it. So far, 180 contributors. A lot of tools and resources put in perspective together. The contributors are experts in research and data management or in scientific domains in ELIXIR and beyond. There has been contribution from EMPHASIS and as well as from other research infrastructure. It's managed by an editorial board composed of 16 ELIXIR members from several nodes and the lead for fair data resource at the Office of Data Scientific Strategy at NIH. So it's already both US and European project. And the content is created by members of the ELIXIR RDM community. And this is a very welcoming community. It's really easy to join it. So there is a plant page, and the entry point is a plant science page that gives a general principle of how you should manage your data. First, the importance of plant biological material, then the phenotyping, data on metadata collection and publication, same for genotyping, as well as related pages. And in all those pages, the different plant data standards are listed, of course, and their different interest is presented. And within the plant data standards, there are different categories, in fact, and we are not, once again, talking exactly about the same thing. First one is the semantic standards. There are the description of the data, the controlled vocabularies, a list of term names and their definitions, on possibly turned into ontologies, which provide on top of controlled vocabularies, semantic linking between terms. A good example are, of course, the crop ontology, and we all know about it here, or the sequence ontology for genomic. The important point for me here is that to make it efficient, is that it must be biologist driven. Then you've got structure standards, which are about formatting and organizing the data, about building data models. They can be very simple standards such as CSV or more elaborated, such as VCF, GFF, and MIAPPE. For it to be efficient, we need to have both biologists and computer scientists around the table. Finally, you've got technical data standards that provide data integration and sharing and interoperability between tools and systems, such as GA4GH for genomic data and the breeding API for plant breeding data. And this is mostly computer scientist driven. For this to work, of course, we need a common glue, which are the permanent, persistent unique identifier. In other words, URI, Gen ID, accessions ID, trait IDs, DOI, and so on. So for the semantic standards, the ontologies, most of the time, the sequence ontologies, the gene ontology, are used to annotate one object, one protein, one gene, a plant anatomy, and so on. Therefore, the concept that are listed in those ontologies are atomic concepts, such as one protein function, one cellular localization, one plant organ. But for phenotype, well, it's more complicated. This is where the crop ontology approach, the triplet, trait, method, scale, came very paramount, very important for the past 10 years, and it has been proven to be a very efficient solution. I'm not going to explain it here in detail because I am assuming that you know it. What I would like to draw your attention on is one of its drawbacks. For instance, here, if we want to talk about the variable plant height, we see that we've got one trait with 4 methods: total height, first tassel branch, last expanded leaf, or because our colleagues from biology are highly creative, highest pixel corresponding to plant. And therefore, you've got finally units, which can be international system units or the number of pixels. When we see this, when we see what our colleagues are doing, we see very quickly that there is an uncountable number of combinations. We really face an exponential explosion of the number of variables. Each trait, method, or unit has to be identified if we want to share and reuse the data. Now, that's a problem because one ontology with more than 1,000 variables is very difficult to manage. But there are solutions. If you want to describe the phenotyping variables you are using in your dataset, in your experiments, the first recommended approach is to go on the crop ontology website to have a collection of species-specific ontologies. You've got also these other ontology portals that take the same approach, but with more community-oriented ontologies. Indeed, we have seen that the methodology of measure can be specific of each community, for instance, for CGIAR and INRAe. Therefore, there is a need for consortium and dedicated ontologies. If this is the case, those dedicated ontologies can be stored in this portal or in any other portal. But ideally, what we recommend is to contribute to the crop ontologies when that's possible. When none of those two solutions are possible, and there are cases when this is the case, the idea is to include the crop ontology triplets, sorry for the misspelling, inside the MIAPPE dataset. Also, to find the variables, the traits that you are interested into within all of those ontology, I draw your attention on this ontology term search engine, the ontology lookup service, available at TBI. So if we talk about including those variables within MIAPPE, so let's have a closer hint of what it is. So it's a minimal information about plant phenotyping experiments. You've got the MIAPPE.org website. It has been built by many stakeholders, ELIXIR and EMPHASIS, Biodiversity, North American PPM. It's an open community with requests for comments, GitHub feature request, mailing list, meeting and work groups. So far, there has been 2 major versions published. There is a third one on the way. It addresses the needs of crops and woody plants, and it has 3 publications so far. It's a minimum information for biological and biomedical investigation standard. It means that it aims mostly at being biologist friendly with clear definition of the examples. And we have devoted a lot of time on building those definitions and examples. It means also that it is provided through Excel templates because that's easy to manipulate. And that we have a lot of focus on training. It lists a minimal and sufficient list of metadata, including the objective of the experiment, who contributed to it, what were the procedures, and what was the biological material, and so on. And the important thing to keep in mind is that MIAPPE is solely a specification. It has to be implemented in different systems. For this implementation, you've got first an ontology on all implementation available both on GitHub and on the Agro portal. It represents the data model. You've got also file archive, ISATAB, and ELIXIR currently under studies. And finally, web service, the breeding API, which means that if you are breeding API compliant, you are in fact MIAPPE compliant, which is a good news because it simplifies things. So the BRAPPE is an international collaboration that aims at providing standard open web service API. The main target is the breeding, but it is being more and more successfully used for agronomy. It is funded, among others, by the Excellence in Breeding platform, and especially the CGIAR, through the commissioning of coordinator, Peter Selby. The MIAPPE specifications are available on MIAPPE.org through the MIAPPE primers, as well as on GitHub as an Excel template. You can also have an extensive explanation in the last paper enabling the reusability of plant phenomics dataset with MIAPPE. This specification table has several sections: investigation, study, persons, the list of data files, the biological material, very important. The other very important one is the observed variable at the end. And you've got also environment, experimental factor, events, observation unit, and sample. All those sections are linked and organized through this data model. It usually takes me half an hour, 1 hour to explain MIAPPE extensively, so I'm not going to do it now. But just to give you a quick overview, the investigation is a whole data set. It can be one field, it can be one greenhouse, it can be a field network, so it is one-to-many location. And since this is crop or perennial plant, it can be from 1 to many years. Each experiment is, in fact, a study. So it's one experiment in one location for 1 to several years. So we've used the term study to describe the experiment to stick to the ISA backbone, ISA meaning investigation study assay. And finally, the assay is a trait or index, the phenotype or environment one, that is being observed, and it follows the trait method scale plus a level. So level being a plant, a microplot, a block trial study. So to use these standards, you've got several data management tools that allow to drive, to enable the data acquisition and management during the experiment. A lot of the time, this is done simply through files. So for this, the template, the MIAPPE template available on GitHub can be used. But to exchange those files, it's good to have a data repository such as Phenome 6 that is being used in the Agent H2020 project. It provides a simple shared space for data as well as standard operating procedure. And it allows to avoid the I've sent my data by email, which is a good way to doom with managing your data. And the other important thing for Phenome 6 is that it's usable beyond phenotyping. But when you've got more than a simple phenotyping network, when you do your measurement by hand, when you begin to have drones, to have networks of sensors, well, when your experiments are fully instrumented, you need a dedicated solution, a dedicated platform information system, such as PIPA, FIRS, the one deployed at IPK in Gatersleben, etc. And for this, I'd like to draw your attention on one upcoming presentation within this series of webinar that will present FIRS, the solution developed at Phenome. Then you've got the data management tool during the course of the experiment, but after the experiment is done, you need a data repository to provide sustainable data access over decades. For this, you've got several solutions. First one are generic data repositories, such as Zenodo, provided by CERN, and that is very strong and reliable at the European level. You've got Dataverse with either the Harvard instance or other instance across the world. And the interesting thing is that you can provide MIAPPE metadata within Dataverse and Zenodo instances. And by doing this, you will ensure that your data are really correctly described and that they can really be reused. For this, there has been a training a couple of weeks ago. The training material as well as the recording are available on this URL. And there will be also linked on TESS, the ELIXIR training material portal, so it will be available there soon. And this training is part of the plant phenomics assembly that is described here, where you see that you've got the Zenodo and the Dataverse instances available here, as well as the fields system for data management. And this plant phenomic assembly is available on the RDM kits. And although if you don't want to use generic data repositories because your data is too big, because one experiment of Phenomics can be tens of terabytes, or because it's too complex to be represented through simple files, you can have tools dedicated to phenotyping, which is the case for FIS, PIPA, BMS, so breeding management system, breedbase, etc. Those systems are complementary to generic repositories. In particular, since generic datasets can be linked to specific database. In other words, you can have in Dataverse a link to one of those databases, FIS or PIPA or so on. By doing so, you've got the best of the two worlds, with both a representation of the complex data and a link available on the long term on the generic repositories. Here I've put an example of this approach. And it can be seen here. So this is a screenshot from a Dataverse where you've got this link to data here to GMPIS. Finally, what I would like to say also is MIAPPE allows to have data integration between silos from phenotyping to genotyping. Indeed, in Phenomics, we've got biological material, field experiments, and in genetic genomics and omics in general, we've got also this biological material representation and identification. And by spotting those two, we are identifying key resources, pivot objects that are shared between the two, and that can be used for integration, for linking, especially if we are using the same standard between the two. To do so, the MIAPPE standard, though initially dedicated to plant phenotyping, has been embedded within biosample at EBI, so therefore available for sequence data and variation data to identify the material source used both in genotyping data and phenotyping data, hence enabling both integration and discoverability in community data discovery portals such as FEDER. FEDER provides a findability global data discovery portal. It indexes MIAPPE sources, breeding API sources, as well as dataverses and other historical data discovery network provided by the WIT initiative. Last slide. The take-home message I'd like to share with you is that data management, it relies on a complex life cycle, and you've got to be careful of what you are talking about when you are talking about your data, because you've got the observation, the measures, but also the derived and the reduced data. And we need to link all those different data sets. We need also to be very clear on all those steps of this data life cycle, ideally through a data management plan. And not all steps of data must be shared, but it is really instrumental to share the raw data sets. And of course, the final data set to back the scientific publication, alongside sufficient provenance to understand exactly how the data has been transformed. All of this work, when it is community driven, and I draw again your attention on the RDM kit, data standards and tools are easier and easier to use each year. We are working very hard on this. Data acquisition tools are often community specific, but they can be reused beyond their original communities. This is the case for FIS, for some CGIR tools, especially the BRAPI compliant one. And regarding the open science policies, sustainable sharing and findability are keys, and be very careful of the data terms effect. It's not because you have published your data somewhere that it is findable and reusable. And this being said, I'd like to thank a lot of people who have contributed to everything that I've presented here.
Thank you, Cyril. Did you finish or did you?
I'm done.
Sorry. I was waiting for the last word. We can see so many names on that slide, so it's really a community effort. Thank you for this very detailed overview of the Phenomic data management tools. I would like to invite our audience to, you can use the reaction button and raise your hand, or if you want to ask a question to Cyril, a burning question, just open your mic. I think you are able to do it on your side. If not, just send me a mail. Anybody having a question for Cyril? Raise your hand. I hope I can see. Perhaps, Cyril, you can stop sharing.
Yeah.
This way we can see. Okay, we can have a better view of with. I have questions, so Pierre can open the floor unless somebody is thinking about having one. First, yes, I have a question on the RDM kit, the European RDM kit, because it's a community-driven platform. You mentioned an editorial board. What is the role of the editorial board? Because if someone from the community raise the attention of this platform to a new standard or a standard, but it seems competing with an already recommended standard, how do you mitigate this situation to decide which standards should be recommended, or are you promoting both with pros and cons? What is the process?
So first, the important general philosophy is that when there are two competing or complementary standards, we promote both. Okay. We try to avoid to have the RDM kit as a portal of a single group. For the editorial board, there is a general editorial board, the 16 people. And when they see requests that are targeting, for instance, the plant pages, they propose a discussion from between the people who proposed an evolution and the initial contributor to the page. Therefore, every specialist of that domain can argue and find a consensus on how to present things.
Okay, so the role is to moderate between two users of a standard. Okay, good. Another question I had or comment is, of course, you can expect that I will talk about the prop ontology. Thank you for promoting the triplets format, which has been, as you said, proven, being used in many databases. The only thing I wanted to say is you have a lot of variables in the crop ontology, but if you look at each trait, I think the maximum of variable you will find will be 5, 6 maximum. So there's no that much method to measure a trait. And even the pixel, we've been approached by people using, of course, image recognition or drones. Even for the pixel, even if we put that method, probably if you don't want to create a variable for the pixel because you want the breeder to really understand what it means, therefore you may just think about having a conversion. So what does the highest pixel tells you in terms of plant height in centimeter or meter? Did you discuss that already in your community? Or?
Not that way. what we did in our community, which is Phenome, is at least to have it's centered around the phase system. The idea was to get everybody at least to list their variable inside phase in the shared public instance of phase.
Okay, in phase.
And we tried, I think, in 2018 to merge what was done in the wheat crop ontology. We have been working, you and I and Marie together on, with what they were doing. And what they are doing is so complicated that it was way more efficient for them to build their own list and to focus on the trait. And the trait, in my opinion, is really the important part where people should be sure to reference either the crop ontology or to have a quadruplet approach with entity and attribute, with the entity picked from the plant ontology and the attribute from the petro. But trying both to get people to correctly describe their high throughput variable in phase and to integrate those lists with those of the wheat crop ontology, while they were trying at least to agree among themselves, it proved too complicated.
Yeah, yeah, because first you had this validation step within the
community.
before. Okay, it's interesting because we have been approached by other research projects using machine learning or doing NIRS, and then have asked us to add a method type for their results. So that's why I was wondering how you debated on that. No question from somebody else, because I feel I'm debating. I still have a comment, but anybody has a question on those points? Or no, I don't see any hand raised. So my follow-up, I have another question is, you said that perhaps I misunderstood. If you are BRIAPPE, BRIAPPE compliant, if your database is BRIAPPE compliant, then you are MIAPPE compliant. It means that all the fields of the MIAPPE metadata standards are included in BRIAPPE.
Yeah.
Okay, so that's great. And if you have because normally a standard shouldn't change, but we know that over time MIAPPE will go through additional versions, so it means you have
so does BRIAPPE.
So you have to follow in BRIAPPE as well, but both are behind the community, so that's good. And my last point is I read the last, or perhaps it's not the last, but the paper from your colleague Paputsoglu. She published a paper, I have it here, called "The Benefits and Struggles of Fair Data: The Case of Reusing Phenotyping Data." She had a point on the fact that there was specification published for developing a fair data point, a short list of metadata that should accompany the fair data you want to exchange. And she was, in the paper, they are, in fact, her and co-authors, suggesting that MIAPPE could be kind of an additional meta module when you, of course, try to verify and put your data in access around phenotyping. So I thought it was it's an interesting idea that probably needs to be developed in another webinar, I would say.
Definitely. Either a webinar or possibly a topic, a project for an upcoming biohackathon. Either one of the national one or the European one or the Japanese one even. But yes, adding MIAPPE specification in fair data point could be very, very, very interesting indeed.
I think
thanks for
it's a suggestion. I will send I don't have the link just with me, so I cannot put it in the chat. But anyway, if you look for a paper called "The Benefits and Struggles."
So, I have that paper in my to read list to be very honest.
But I wanted to share with our participants today. But people, you can look for the DOI. It's the benefits and struggles of FAIR data, the case of reusing plant phenotyping data by Evangelia Paputsoglu and with the 3 other co-authors, among which Richard Finkers. So anyway, I think it's worth being considered. Another probably thing we should discuss in the future is MIAPPE is of course the standard for the phenotyping experiment, but not all the phenotyping data are coming from experiment as we think an experiment. I mean, something which is driven by a scientist on an experimental station. Now we have an approach for on-farm data, where there's less control of the parameters, environmental or management. So is this something you've been approached for MIAPPE, or it's outside the scope of MIAPPE?
So currently it is outside the scope. It is something that has been discussed as a challenge in some part of the project I'm involved into. I'm sure I'm not the only one. Currently, I personally don't have a clear enough understanding of the challenges of on-farm data acquisition to have any position on that. But there is more and more crowdsourcing data and on-farm data being gathered. Definitely, this is probably one of the next big things that should be taken care of.
Yeah, I think so, because we do have a platform for this type of crowded, I don't know what to say, on-farm data. And we have been already, at least the developer of that platform has been discussing with MIAPPE community already. But I don't know if we talk to MIAPPE, do we talk to MIAPPE as well? It should be both, I think.
It should definitely be both, and with connections to some people in Europe. Yeah.
Good. So perhaps a topic to add to a next PhenoHarmonies meeting.
Definitely.
Okay. So I think this was my last comment. Nobody else would like to open the mic and ask something? Mathieu, please open your mic. I hope you can. Otherwise, I will do it for you.
No, I think I can. Can you hear me?
Yes.
Yes.
Okay. So thanks, Cyril, for the presentation. You discuss a little bit about interoperability between phenotyping data and genomic data, mostly using DOI. I think you are referring to the DOI that you can get with GenBank accession, for instance. What would be your recommendation when we don't have any DOI on material, like material coming from breeding programs, where you don't have really a standard to identify uniquely the material?
In all situations, whether it's lab or breeding program material, or even a public catalog variety coming from a seed company, we always have this triplet that we find in the MCPD, an accession number or any kind of ID within a holding organization and possibly for a given species. You should, in your lab, in your seed company, have those 3 elements somehow present and concatenate all the 3 in a URI. This is, in fact, the approach made in Phys, in Phenome, where the varieties are proposed with a set of URIs built with the seed company / species / a number or name or whatever has been picked. But the important thing is really to be sure that you've got the namespace, the name of your lab, something that you know will remain unique for the 10 years or later. But this is a case that we all have And definitely the DOI is a solution for gene banks, but it's not the solution for everything.
No, yeah, I agree. But then if you have your plants that are phenotyped in one organization and your genotyping in another organization, you will have different identifiers. And it will be difficult to cross. And I was wondering if we don't have a kind of intermediate layer that could help to make this linkage. Like, I don't know, maybe you refer to biosamples, or is there anything that could help to make this link, this connection between objects that are identified differently, even if we are talking about the same material?
So one thing that we have seen very important in a lot of projects is being able to carry synonym IDs in biosample or in any other database. In your genotyping sample, having the information that your material source Geno 123 is in fact corresponding to my partner lab source ABC, for instance. Having both of those helps. It doesn't solve everything, but it helps. And although at a consortium level, we should somehow agree. There is no magic trick so far. And finally, something that is done, and this is what you mentioned also, is to pre-register very early your material at biosample, even before it gets genotyped. When you know that you are going to do some integration between omics data and phenomics or whatever data, registering your samples at biosample before even beginning to do the actual sequencing, and then keeping this biosample ID as a traceability with all of your sample, with all of your data, is something that will definitely help. It will not solve everything, but it will solve a lot. This is what we are trying to do in some of the national projects.
Okay. And these biosamples are the same as the one used in NCBI, or is there any synchronization, or it's different?
So those are different databases. So
Okay.
between the Japanese one, the European one, and the NCBI one, they are synchronized, but it's always a one-way synchronization. In other words, if you are creating at NCBI your biosample, its information gets sent to EBI. But if you want to do an update of the metadata, you must do it at NCBI, not at EBI. It makes sense, of course. And also, I'm not sure that all the metadata get synchronized and transported, because since we have added some MIAPPE metadata at EBI, I got the impression that some of it got lost when transferred to NCBI, but not 100% sure.
Yeah, but at least there is some synchronization mechanism between the two.
There is some synchronization mechanism, but at least within a consortium, within a community, I would advocate pick one and be sure that the whole community stick to that one, either NCBI or EBI. But if you've got part of your sample in NCBI, other part at EBI, and last part in DDBG in Japan, it's going to be messy.
Cool. Okay. Thank you. Thank you, Cyril.
Thank you, Mathieu.